think we can get started. So I'm going to read this disclaimer statement, and then we will introduce our guests today. The information and opinions shared by our panelists will be generalized statements and are not intended to be interpreted as a promise for future treatment outcomes or a specific timeline for such outcomes. While we are all eager for advances, it is impossible for anyone to predict when a treatment may become available or anticipate roadblocks. This session is being recorded and will be shared on our public YouTube channel after the conference portal closes on August 31st. You'll also be able to rewatch a recording of this session in the next couple of days within the conference portal. Okay. Great. So let's start with introductions. Professor Erdmann, would you like to start?
Yes, thank you, Rachel. So my name is Jeanette Erdmann. I'm from Germany. I'm a professor for genetics, especially cardiogenetics, normally, and I'm a patient with collagen 6. And since I made my diagnosis a few years ago by myself, actually I started to work a bit on a therapeutic approach. I'm having a glycine mutation in collagen 6A2, and we're trying to use antisense oligonucleotides or CRISPR interference to overcome this mutation.
Great, thank you. Professor Dumont.
Thank you. I prepared a few slides, so I'm just going to share them, but I won't take much time. It's just you.
Oh, okay.
Yeah, we were going to do introductions first and then slides, if that's okay.
Oh, you do just an introduction? Okay, sure.
Yes.
So I'm Nicolas Dumont. I'm an assistant professor at the University of Montreal. We're focusing on muscle diseases, especially how muscle stem cells are affected in these diseases. And we have a very strong team of collaborators here to help us kind of dig into the mechanism of different rare disease. And we got a special focus recently on collagen 6 myopathies. So we're trying to use the new technologies that we have to kind of understand better the disease and try to find some potential treatment for personalized medicine.
Great, thank you. Professor Allamand.
Hi, everyone. I'm Valerie Allamand. I'm a researcher in France most of the time, and currently in Sweden on sabbatical at Lund University. And I've been working on congenital muscular dystrophies for a long time now, and collagen 6 for about 20 years.
Wonderful, thank you. Gustavo?
Oh, I think everybody knows me. I'm the scientific director of QCMD. So learning and helping move forward all these great scientists to achieve clinical trial readiness and one-day treatment.
Dr. Foley?
Hello, I'm Reghan Foley. I'm a pediatric neurologist and neuromuscular specialist. I'm based now at the National Institutes of Health working with Dr. Carsten Bönnemann, and I've been a collagen 6 focused clinician and clinical researcher probably since 2007 with Dr. Bönnemann, and in my time in London with Francesco Muntoni, Professor Muntoni, and now I'm still cheering on the collagen 6 patients in the community.
Wonderful. Professor Muntoni?
Well, I'm a pediatric neurologist. I work in London and at the Institute of Child Health at Greater Ormond Street Hospital. My research is on rare neuromuscular disorder and on antisense and oligonucleotides, mostly for Duchenne, but I'm lucky to have a very good collaboration with the group at NIH, with Carsten, and with Carsten I co-supervise a shared PhD student between NIH and UCL. That is I think is a new initiative, and I'm grateful for this initiative that allows really to do collaborative work.
Great. Dr. Bönnemann.
Carsten Bönnemann. I'm also a pediatric neurologist, neurogeneticist at the NIH, interested and involved in early onset muscle disease clinically and in the development of therapeutics, and collagen 6 is really the core disorder that we're trying to address and find ways to treat.
Great, thank you. I believe you have some slides to share first.
Yeah, I want to just
give like I did in another section, a bullet point list of considerations that I think are special for collagen 6, and that link us a little bit back to the morning session as well. So this is not an overview of collagen 6, just the few special things about collagen 6 that we need to address. So collagen 6 and 12 for that matter as well is part of the muscle extracellular matrix. It helps organize the supportive environment of muscle cells. Its absence or dysfunction has complex consequences affecting the maintenance and health of muscle fibers that are surrounded by collagen 6. However, it's not produced by the muscle fibers, but it's produced by special cells in between the muscle fibers that are interstitial cells that we call the FABs. And that'll be interesting later. It's also important in other connective tissues, and notably tendons, skin, but maybe also vessels and other connective tissue-rich tissues in the body. There are 3 disease-relevant genes for collagen 6, one of which is very large and doesn't fit in AAV. For instance, COL12 is also very large and doesn't fit in AAV. Importantly, there are 2 general types of genetic mutations, and that's true for both 6 and collagen 12. One is recessive loss of function, leading from complete loss to diminished function, and the by numbers more important mechanism is dominant negative mutations. And they come in 3 flavors: exon skipping mutation, glycine missense mutations, and a newly discovered pseudoexon insertion that I alluded to this morning a little bit. Just to show you a crude picture of what I mean by muscle extracellular matrix. So this is the muscle membrane. This is the muscle membrane surface, and down here is this very highly complex network of molecules that collagen 6 is part of, and is also involved in regulating signaling molecules and other things that need to be presented to muscle cells to support them. Collagen 6, as I said, is 3 genes with both dominant and recessive mechanisms of disease causation. And this is an illustration that collagen 6 is actually not made by muscle fibers, which are here in red, but by muscle supporting interstitial cells that are shown in green. And you can see the collagen 6 reaching out and making that contact. And that's important because these are the cells that need to be genetically targeted. And then lastly, a point about this dominant negative mutation mechanism. It means that if you have a mutation on one of the 2 copies of either one of the 3 genes that make collagen 6, and these mutant chains are allowed to form the collagen 6 and participate in the formation of collagen 6, then you have a dominant negative effect, meaning that most of the collagen 6 has at least one of the mutant chains on board and is poisoned by it. And most of the mutations are sitting in one particular spot of the collagen 6 genes. And one therapeutic approach done is that Professor Erdmann is also mentioning, is to inactivate a mutation early on so that it's not allowed to incorporate. And then you have 50% normal of collagen 6, which fortunately is enough to prevent disease from forming. So that's all in terms of background that I wanted to share.
Can I ask you a question real quick? You said that it's the mutations aren't targeting the muscle cells themselves directly. So if that's the case, why do you know why we have muscle weakness?
Well, that's a really relevant and good question, and something that is really insufficiently explained. There is we call that a non-cell or not autonomous muscular dystrophy. So the genetic defect is not expressed in the muscle cell, and yet the muscle cells suffer from it. And so there are different ways why that can be. Regeneration can be impaired because of the regenerative environment being different. The signaling to the muscle cell by all the supportive growth factors that are regulated by the matrix is insufficient. So the muscle cell sees an unhealthy environment that's not properly regulated. And there can even be structural consequences of having an abnormal matrix around it, which then has consequences in the muscle fiber itself, including differences in autophagy and apoptosis and all of that. But it is really the covering the surrounding that keeps the muscle fiber healthy. And if that goes away, then the muscle fiber suffers, even though it doesn't have the genetic defect.
Okay, thank you. Professor Erdmann, would you like to share a little bit about what's happening in your lab?
Oh, yeah, actually. So I'm totally, no, I mean, how should I start? So actually I'm relatively new in the field of new genetics. So my main scientific focus is cardiogenetics. So this is obviously, I'm interested more in complex diseases, but with having my own diagnosis, which was actually only at the age of 45, I'm more interested in also in rare diseases, monogenic ones. And I then started to establish a small working group in my institute to work with actually a therapeutic approach targeting my own mutation. So I have now 2 PhD students, both are sitting in the audience, by the way, and they present posters. And we are trying to establish two different methods. One is the antisense oligonucleotide method. So we want to suppress the mutant variant, as Carsten Bönnemann just shortly explained. And the other mutation method is to use CRISPR interference. So the idea is to silence the mutant allele, and hopefully, that will lead to the 50% of good collagen 6, so that might help not to develop the phenotype.
Great. Yep. And as she mentioned, there's a poster in the poster section of the portal, and there's actually an audio file from one of Professor Erdmann's students. So it explains the poster. Professor Dumont, would you like to share some slides?
Yeah, sure. Everybody sees the slide? That's full screen, no?
Yes.
It is? Okay, there we go. So just briefly where we are and what we do. So our lab is located at Saint-Justin in Montreal. This is the largest mother-child hospital in Canada. It is associated with the University of Montreal, and this is our team. Well, the picture was taken about 2 years ago when we can all fit in the same 2 meters in the same frame. Hopefully we can do that again soon. And basically the focus of the lab is not that much on muscle fibers, on muscles, but more on muscle stem cells. So basically in your muscle, you have the muscle fibers that are responsible for contraction and movement. But you also have another cell type, which are the muscle stem cells, and they're responsible for muscle regeneration. So here you have an example of a video where you have muscle fiber, and you have right here at the bottom, a muscle stem cells that will get activated after an injury, and eventually will start to migrate around and will start to divide exponentially the first hours after an injury and generating a lot of cells. And these are these cells that are responsible for muscle regeneration that will eventually fuse to the muscle fiber and repair the damaged muscle fiber. And what we now know is that in a lot of diseases, these cells are defective. So they're not functioning as well as they should be, and they're not regenerating the muscle as well as they should. So we are focusing on trying to address what are the defects in these muscle stem cells, and how can we target these cells to really improve muscle regeneration. And we do that by using new technologies. So basically, we're using the induced pluripotent stem cell technology, which is relatively new in the field. It's a technology that allows us to take samples from a patient, usually blood cells, and reprogram these cells into stem cells that we can then differentiate into pretty much anything we want. And in our case, we are differentiating these cells into muscle cells. So the idea is to take cells from patients, generate the stem cells, and then differentiate into muscle cells that we can study in a dish, and especially with the goal of trying to find new drugs. So here we have myogenic cells from collagen 6 patients, and we generate 3D muscles out of these cells. So we're kind of recreating a small muscle in a dish from the blood cells of the patients, which is kind of new, interesting technologies that we can now do. And we are putting different drugs on these muscles, and we can see if the muscle grow better and regenerate better and contract better. Here we have just a small video of the muscle that will be contracting and then getting very green, so that when it contracts, we can follow the contraction and see if the treatment that we put on these muscles is kind of improving the regeneration. So the idea of being very to try to find some kind of personalized medicine for a specific patient that would improve his muscle, basically. So that's pretty much it.
Fantastic. Thank you so much.
Beautiful images. And I always have a question for you regarding your 3D model. Do you have also the fibroblast producing collagen 6 in your 3D model, or is pure muscle cells?
That's a very.
Is that important or not?
Yeah, exactly. We did not do co-culture with fibroblasts yet. We will. So the idea being that we can put fibroblasts in and try to see if we can kind of rescue their expression of collagen 6, because they're the main expressing cells. We do add collagen 6 in the dish to see if just the presence of collagen 6 could rescue the function of the cells. So that's something that we will do soon, also to do co-culture with fibroblasts. Yeah.
Thank you.
Great. Thank you. Professor Allamand.
Let me share my screen. So I think, I'm not sure if I can share already.
There we go.
So, like I said before, I'm usually based in France, and I'm currently in Lund. So I'm also working a little bit on Llama 2 now. One thing that we know is that homozygous nonsense mutations lead to complete collagen 6 deficiency, and those are recessive mutations. And like Carsten Bönnemann said earlier, there's definitely an interest in targeting the primary defect, so the mutation itself, in order to have the most functional benefit or therapeutic benefit. So that's why I'm interested in recessive mutations, and in particular nonsense mutations that introduce PTCs or premature termination codons in the sequence. And basically, the goal is to target these mutations in order to restore a functional collagen 6 in the extracellular matrix. And the models that we use are either cells from patients, dermal fibroblasts, and also for some of them muscle fibroblasts. And we also developed a few years ago, or several years ago, a mouse model with a nonsense mutation. So we have an animal model for these type of mutations, and we use pharmacological strategies, so drugs that will bypass the premature termination codon. That's the process called read-through. And we also associate these drugs with molecules that will inhibit the degradation of the messenger, the transcript, in order to be more efficient. And also there's one strategy that we are developing that is a gene therapy strategy, but not using an AAV, because we've tried that with one serotype and it did not target the muscle fibroblasts. So we are using another vector type to do that. So hopefully this will target better the muscle fibroblast, and we will get some rescue there.
Thank you. Okay.
And maybe
Dr.
just-
Bönnemann, Dr. Foley, do you want to talk about your work that's happening in your lab?
Yes, hang on. Oops. Okay, just briefly to tell you what the three major research focuses are preclinically in the lab. So one is to investigate the consequences and therapeutic opportunities of the altered function of the matrix, and in particular on growth factor signaling in collagen 6 deficient muscle. And we use a collagen 6 deficient mouse model for that to see whether there's any therapeutic avenues to support muscle. Then we do mutation-directed therapeutics, in particular precision therapeutic approaches against dominant negative mutations for the exon skip mutation and the glycine missense mutations using siRNA as our primary tool, but also CRISPR-Cas and base editing. And then importantly, a splice modulatory approach to the pseudoexon mutation that I described also this morning in very close collaboration with Francesco Muntoni's and Haiyan Zhu's group at UCL. And then also in terms of tools and technology development, we have generated a humanized mouse model for preclinical testing of these splice modulation and other approaches that really has the relevant part of the collagen 6 gene exons and introns completely human, so that our therapeutics can be directly translated. And then very importantly, we are working on effective targeting of these collagen cells of origin, the Fabs in the muscle, for ASO siRNAs, but also developing and finding new AAV serotypes that are capable of going into the Fabs to enable both gene therapy for COL6A1 and A2, but also to deliver other therapeutic tools such as CRISPR and U7 approaches to the appropriate cells.
Great, thank you. Professor Muntoni, is there anything that you would like to share about what's happening over in London?
It's raining, but apart from that, I think the also in collaboration with Carsten, we are looking at one of the bottlenecks of antisense oligonucleotide is the targeting of muscle interstitial fibroblasts, and I think we have some projects on that as well. And we also have, this was almost an incidental kind of work we have done on transcriptomic analysis of patients with different level of clinical severity, both fibroblasts and muscle. And this is gave us some interesting insight in like the complex pathogenic complex step that occur once collagen 6 is dysfunctional. Those are the main things we are doing, and I think that we are targeting many of the, well, we're working with Carsten to target that particular collagen 6A1 common mutation, and also we are trying different technologies and different antisense chemistries to try to identify, for some of the dominant mutation, you'll find that it becomes highly personalized, and what works for one particular mutation may not work for another one. So we have somebody working with what the colleague Carsten mentioned, Haiyan Zhu, who is trying to develop, trial and error, honestly, of which are combination of backbones of the different antisense that may achieve, especially for the silencing, the more selective silencing. But I think we have, maybe we should say once a month, once every 2 months, I don't remember now, collaborative calls with Carsten and all his group, and we share all this learning. And the new combined PhD student is putting a lot of glue in between the two labs, and at the moment he is based exclusively at NIH because of the COVID restriction, and we're not even sure where we will ever want somebody from the States unless fully vaccinated. And I'm joking, but at the moment there is a little of hysteria regarding who we will let in or not in. So those are probably the main things I'm doing, and I'm interested now because there is can I ask a question?
Of course.
Yeah, so I'm interested because one of the colleagues, Jeanette, I hope you don't mind, I'm calling you by first name. So you come from the cardiology field, and of course, an interesting observation, coming back to some of the point that was made also by Carsten, is that regarding the function and the complex pathogenesis of collagen 6, but one that I think is a good one is that cardiomyopathy really does not appear to be a significant problem. So that must be something quite unique of what collagen 6 does in skeletal muscle compared to cardiac muscle. But I'm interested to have your insight into that.
Yeah, so actually this is something that makes me also happy as a patient, that the cardiac muscle seems not to be involved. However, we know from our GIVA study, so my main interest is coronary artery disease and myocardial infarction. And I have done a lot of genome-wide association studies, and actually collagen 6A3 is a risk locus for coronary artery disease. So we don't know the exact mechanism there, but it pops out as one signal, strong signal. So there might be something that reaches from the neurogenetics field to the cardiogenetics field from collagen 6.
Okay. Question answered. All righty. Do we want to go ahead and move on to community questions then? Is that good for everybody? Okay.
Yes.
Awesome. So I would like to touch on Abe's question first, because I think it's pretty deep and may take a little while. Can you comment on the turnover of muscle fibroblasts and whether gene therapy with AAV might be durable? And 2, any thoughts on manufacturing COL6 trimers, which could then be delivered IV? Something similar to what Prothelia is doing with LAM-111. I'll open that up to anybody.
Seems to be directed.
That's a big question.
Yeah. It's a good question. So we don't know what the turnover of these particular cells is in muscle, and it probably completely depends also on the state of the muscle. So if you have a quiescent state in the muscle, then these fibroblastic adipogenic progenitors that make collagen 6, and that's pretty clear just from single cell and single nuclear RNA-seq experiment out of muscle that this is one of the major populations to make collagen 6, in addition to tenocytes, which also exist in muscles and also fibroblastic cells. And in the quiescent state, these are probably not very kind of proliferative or turnover happy. However, in the diseased stage, that may change. So when they get engaged in fibrotic processes, in transforming into myofibroblasts and other more pathogenic cell types, their turnover time probably increases. And so the question of durability of an AAV correction of these cell types is a good one. So hopefully, if we're talking about a gene editing approach, then we wouldn't have that problem because the progeny of these proliferating cell has inherited the genetic correction. If we, however, talk about an AAV-mediated simple gene replacement approach, like is possible for A1 or A2, then the episome would not be inherited if the cells get very proliferative afterwards. So then we can only hope that the gene therapy will dampen the proliferative drive down, so that this becomes more durable, but also think about redosing, obviously. The turnover of the matrix itself is very slow. So once this is deposited in the matrix as a collagen, that turnover is much slower. So that's an approximation to your question, but that's somewhere where that is playing out. In terms of a protein therapy, probably at this point in time, this is prohibitively large. So the functional unit is the tetramer, and the tetramer is the size of a eukaryotic ribosome. So that's a huge chunk of protein that you would have to get into a diseased muscle matrix by systemic administration. So I don't put much hope into that approach. I have to say the panel may disagree, but I think going for genetic correction is a much more hopeful approach for that. I'll stop here.
Anybody else have anything to add to that? No, we're good. Okay. Dr. Bönnemann, your AAV project, the related question here, is there a situation, an outlook on vectors that are able to deliver iRNA or ASO to fibers?
Yeah, so that the purpose of finding things that target is not just for AAV gene replacement therapy. Even more importantly, it's to deliver the goods, the tools that are necessary for genetic correction. That may include CRISPR-Cas, that may include base editing, that could include transcript editing, and could also include the directed delivery of silencing tools, such as siRNAs that are read off the vector, or exon skipping tools that are read off the vector with the U7 promoter. All of that will become feasible if we have an effective tool of targeting the Fabs. In addition to that, Francesco's group and our group are also working on just making simple ASO go to the Fabs. And that probably requires peptide modifications, chemical modifications, lipid modifications. It is all, we have the tools to screen for that. We have the tools to look for that. So it's a matter of time, in my hopeful estimation, that we find something that is effective going there. No one has looked for that. So all muscle-directed therapies were always kind of looking to target muscle cells. And so maybe that's in someone's drawer already and has been ignored. Hopefully that's the case.
Okay, thank you. Dr. Foley, can you give us a status on Omega Pill and whether or not there are any clinical trials slated for collagen 6 for 2021 or 2022?
Yes, so my colleagues who were on our LAMA2 breakout session had heard this, and I'm gonna it's a very important question to answer, of course. So we had been the single site for the phase 1 trial for safety and pharmacokinetics of the oral compound Omega Pill between children ages 5 to 16 years, both collagen 6-related dystrophies and LAMA2-related dystrophies. And the outcome of the study was the compound was safe and had a favorable pharmacokinetic profile. The next step was we had hoped to go on to a phase 2 study, and the sponsor brought their plan forward to the Treat NMD's advisory committee for therapeutics, or the TAC committee. And they came back with some suggestions that are very important, one of them being identification of biomarkers that could be measured if a subsequent trial pursued a phase 2 with the Omega Pill to see how we're affecting those biomarkers, which we do not have currently in collagen 6-related dystrophies. And it's also a recommendation for further preclinical work in collagen 6 mouse models, because we don't have as much data in the mouse models for collagen 6 using Omega Pill. And so we know Omega Pill is safe and has a good PK or pharmacokinetic profile, and it is something that is being still developed and ideally would probably be used in combination, a kind of combinatorial approach with other approaches that are much more transcript directed, as Dr. Bönnemann talked about this morning.
So Omega Pill is a downstream treatment, correct? Could you kind of describe what that means and how that differs from other therapies?
Of course. And so this morning's discussion, we talked a lot about identification of the genetic cause of the transcript being affected by a mutation in the gene. And that's kind of going really to the root of the cause and trying to correct the protein at that level. In contrast, the downstream effect would be the effect of the genetic mutation on the protein may have secondary effects that are not the primary cause, but a secondary effect. And in the case of the Omega Pill, it was acting on what we call apoptosis, or a tendency or proclivity towards cell death. And so it is applicable for multiple congenital muscular dystrophies, namely the collagen 6 LAMA2, but does not go to treat the core cause of the condition. So again, still something very much in our toolbox of potential therapies, but not going to be probably used as a standalone therapy, but more probable in a combinatorial approach.
That makes sense. Okay, great. There's a question about contractures and research being done on contractures. I know Professor Bernaldo is not here with us today. Is there anybody who could comment on kind of the work and are getting a better understanding of what causes contractures, and will new trials target contractures specifically? I'll open that up to anybody.
Yes, I can mention that we have granted this project by Stefano Sparsoni in Italy to start looking at this problem, and He has published a paper out of that fund that we QCMD gave him. We can put it probably in the reference to that paper. But basically, they were trying to figure out the molecular basis for the contractures in collagen 6. What are the molecules involved in the contracture aspect there, working on tenocytes, and so for now it's very basic what we learn, and we need to discuss how to move forward from there, and how to still push and keep going in that type of research that is very important for the patient.
It certainly may be a multi-site process. So the contracture may be in the tendon, in the joint capsule, but also in the muscle significantly. And one shouldn't, hopefully, and tenocytes, as I mentioned before, are very happy producers of collagen 6. And we know from electron microscopy on the mouse model, even though the mouse doesn't have prominent contractures, unfortunately, to go after preclinically, but they have a very different elastic fiber content in tendons. So there is something happening in tendons, but also, of course, in big parts in the muscle. So whatever a therapeutic approach one takes, one has to be sure that most cell types that make collagen 6 are targeted with this. So an effective fibroblastic transduction should not just be fibs, but also tenocytes, of course, to do this.
Okay, thank you. And my error, Ronaldo. We're zoning that, Ronaldo. We have a question about the use of cyclosporine A. There was a publication out of Italy, I believe, on the use of suggesting that cyclosporine A using as early as possible, where the diaphragm is less compromised. Do you have any thoughts or comments about that? Again, whole panel.
I'll start. I'll say, well, as for everything from COVID-19 to vaccination to is a risk-benefit analysis. And I think that cyclosporine is a drug we use in clinic. That is a drug we use with a lot of monitoring, is associated with potentially quite a significant number of adverse events. And it may well be, and there is, carcinogenesis in long-term. If you take cyclosporine for a decade, there may be a risk of tumors and so on and so forth. So I think this was an interesting observation. But I think that and I think the proof of concept, I think, is interesting and needs to make us think. But at the same time, cyclosporine is really, really a sledgehammer that one would not use for a very long treatment. Is likely that the long-term adverse event related to cyclosporine on kidney, on other aspects, will outpace any potential benefit. So I personally would be very reluctant, but I'm happy to hear what my colleagues feel.
Anybody else have any comments on that?
Just kind of just say, reiterate what Professor Muntoni just said. There's two actions of the cyclosporine. One is that inhibition of cyclophilin D, and that's what the activity that seems to be decreasing the tendency of the cells towards cell death. There's also calcineurin inhibition, and that's the immunosuppressive effect that's really too dangerous. As Professor Muntoni said, it's really, the risk is too high to use that. And so if we could, garner the type of effect from the cyclophilin D, which had been, as Dr. Baumann knows too, and Professor Muntoni, had been the goal of the Debio work that had been tried in the past, but had not gone forward, right?
Okay, so the take-home message is do not run out and start taking cyclosporine A, please. Got it.
Correct.
Okay. And we have another question about the relationship to collagen 6 and the neuromuscular junction. I guess there was another paper published on that recently. Has there been further research established on that connection?
Yeah, there was a paper in the mouse model in COL6A1 knockout mouse model that showed some differences in neuromuscular transmission in the mouse model on electrophysiological testing. We've not been able to see that as clinically meaningful part of the disease in humans. That's what I can say.
Okay, great. So we're a little over and we're gonna try and close out here. And I'm gonna ask you all to give one more statement, a closing statement, forward-looking, maybe hopeful for families with collagen 6. And I would like to incorporate one question, if you can, thinking about maybe the top couple of things you may have learned about collagen 6 since we last kind of gathered in 2019. So we'll start with Professor Dumont.
Yeah, sure. So I think that one thing we realized with collagen 6 myopathies and with a bunch of other muscular diseases, that they're kind of more complex than we thought. So it's not just muscle fibers that are affected. We now realize that lack of collagen 6 affects other cell type, and I'm focusing on muscle stem cells, but we just talked about the neuromuscular junction, and although it might not translate into human compared to mouse models, but I think it's a complex disease. But this knowledge about the mechanism of the disease clearly opened the way to new therapies and new avenues that were not explored before. And I think that we can see, based on the panel that we have here today, that there's quite a lot of different approaches that are followed and explored, and with all these different avenues, we kind of increase our chances of finding something meaningful, and especially with the strong increase that has been in the field recently with new technologies in sequencing, in transcriptomics, in 3D modeling. I think we are clearly heading the right way, and there's clearly, I think, hope in the future.
Great. Professor Erdmann.
So yeah, I hope that for myself that I can get a little bit of standing also in the new genetics field. So I hope to improve my knowledge in this field, and then try to translate this into something good for the patients. But another thing what's interesting for myself, and maybe also for all the other patients, is to have a more focus on all the maybe late onset risks that may come with collagen 6. So we are mainly focusing on the weakness, the muscle weakness, and maybe also the breathing issues, but maybe there are more risks that may become only visible in the older patients. So this is something that I'm also, from a very personal perspective, I'm interested in.
For sure. The happy side effect of better standards of care is that we are all living longer, and that means we need to start figuring out what are we doing in our 40s, 50s, 60s, right?
Yeah,
sure.
That's
great. I'm right there with you. Professor Allamand.
Yes, well, first of all, I think that everything that was said today shows that we're all working on similar or somewhat different, but definitely complementary approaches. And I think that's the key to success, because I don't believe in one cure, one universal cure, really. So there are many doors that are being opened, and I'm sure that at least several of them will lead to some therapeutic benefit. So I'm very hopeful. And what I learned is every time we open a door, there are 10 corridors behind for COL6. It is complex. I also definitely learned that we need better models, both cellular and also animal models. They all are very useful, but there are some pros and cons to each of them. So I think, we are still in business for a while, and hopefully with good outcome.
Wonderful. Thank you. Dr. Foley.
Sure, I would say from two different perspectives. From the perspective of the field as a whole, I'd say the patients are the extraordinary strength of this community and are incredible inspiration for all of us. And tied into that from the research perspective, which the patient community has supported, incredibly well, it is so rewarding to be able to review grants both for Cure CMD and the University of Pennsylvania's Rare Disease Orphan Disease Center, and to see every year so many more applications for collagen-6 research and really great translational promise and new people entering our field, young people entering, and it's great to expand as Professor Allamand said, to have different approaches and work together. So I think it's really exciting to see, it's a very collaborative community as well, but to even see new colleagues entering and more applications for grants has really been, really great to see.
Great. Dr. Bönnemann.
Yeah, and I think I said in a different session, but it applies here as well. I think this is an extraordinary time of discovery of tools and methodologies in genetic medicine that almost immediately apply to many of the problems that we face in collagen-6. So that means, fortunately, we don't have to work out every single one of these technologies within our community. We can really take advantage of what's being developed and apply it to our problems and mutations. Having said that, though, at some point we need to take one of them and really go all the distance and go into patients with that. And I think we're tantalizingly close. I'm really very hopeful that we see the first steps of translation of our genetic approaches into patients very soon. And we all hopefully will benefit from that, learn from that, and most importantly, the patients will get better. That's the hope behind it. And secondly, I'm really, sometimes things happen really quickly. And I think the pseudoexon mutation that we've discussed is one example. It's a mutation that we didn't even know existed until a few years ago. And just with the breakthrough in genetic diagnostic technology, we found this, and it's now one of the most common of the collagen-6A1 mutations with a treatment approach delivered on a platter. You didn't even have to think about it. It was right there with the discovery of the mutation, the treatment approach. So we have to be sure that every single patient in our community has firm and clear diagnostic answers in their molecular makeup. That's really critical and key.
Excellent, thank you. Professor Muntoni.
Well, I think that I don't want to repeat what people have already mentioned. I think that one aspect that he wasn't mentioned specifically, and actually Carsten himself has experienced, is it was alluded to, but is the fact that if tomorrow there will be an AAV for fibroblasts, the fact that this is a non-integrating vector will pose some challenges. Having said that, there is expertise that has been developed for completely different disorders, regarding the possibility of using immunosuppression, not cyclosporine, but immunosuppression to allow AAV re-administration. So I think that the bottom line is that learning from a number of different fields will really apply also to this group of condition. And I think that I also agree with Reghan. This is a community that in actual fact is the only neuromuscular group of, together with Duchenne, that really asks me on a regular basis, when is that we do the next webinar? And so I'm going to pester some of you because they've just been on my back, understandably, just this week. And I said, oh, I will certainly do something soon. So I will be pestering some of you for arranging. When I say they've been on my back, I don't mean in a bad way, but I just mean, but they are interested, and we are interested in interested people.
Collagen-6 community is nothing if not interested and aggressive.
Aggressive is not the right
Okay.
term. They're friendly and persuasive.
Yes.
A different A word, Rachel, articulate.
Okay, but we also tend to be in your face about what's happening, so.
In the best sense.
We like that.
Good, good. Keep it up then, community. Gustavo, do you want to wrap us up?
Just a big thank you to everyone, and echoing what everybody has said already, but this diversification in approaches that and we at QCMD we are all the time trying to to fund as much projects as possible and diverse, because as Valerie mentioned, it's probably there is no one cure for all, and we need to go in different fronts at the same time.
Very good. Well, thank you so much, panel. Thank you, audience, for joining us. This recording will be available within the portal in the next couple of days, so you can go back and watch Dr. Bönnemann and look up every word he said in the dictionary. So no worries. So thank you so much for your time today, and we'll be seeing you all tomorrow.
Thank you.
Sure.